So, okay, so I'll tell you a bit about SEC-SAXS as a technique, and also mainly how you can analyze your data, because that actually turns out to be kind of the tricky part. And I'm going to try and do some live demos as well. We'll see how that goes. Okay, so where do I come from? I'm at Cornell. I'm a research associate in Nozomi Ando's group. And this is the group sort of fairly recently. I'm only going to be there for another couple weeks. After that, I'm moving over to CHESS to be a staff scientist, so I'll be joining Richard there, primarily doing crystallography. So, and for the demos, I'm going to show you using raw. So Jesse Hopkins, who's been developing raw most recently, is a good friend, and anytime we come up with new methods, he makes a nice GUI for it. So I'll be demoing some of those. And yep, hopefully you've all downloaded raw already, and you can try these. All these data sets that I'll show you are part of the raw tutorial, so you can play with them yourself. Okay, so a basic outline here. The first part of my talk is kind of the main thing about SEC-SAXS. I'll give you an introduction. So what is that? What is SEC-SAXS? And then even for those who haven't done chromatography before, also just some general principles of what size exclusion chromatography is doing and why it works. And I'll take you through like a basic analysis that you will do. And then I'll also show you some more advanced tools, including singular value decomposition and another one called evolving factor analysis. And then at the end of that, part one, I'm going to just show you very briefly an overview of like some more advanced things that you can read about in the literature. People are doing with SEC-SAXS to really push the envelope. And then in part two, this says part one, should be part two. I'm going to tell you about another technique that's similar that we've been developing in the lab called Regals. It's a kind of general SAXS data analysis tool, but it is particularly powerful for chromatography coupled SAXS data. And I'll show you how we've used it to analyze anion exchange chromatography coupled SAXS, where you have like a changing salt gradient during elution, and also how we've used it to analyze titration data. And depending on how much time we have to spend, I'll show you some demos of those as well. Okay, so first of all, what is the experiment? So we have, basically we have our chromatography setup coupled directly to a SAXS beamline. The data you get is a continuous time series of SAXS data. So you have intensity versus time versus Q. Zooming in on the setup here. Typically, this chromatography setup consists of a pump where you're pumping buffer continuously. You have a sample injection loop, you have a column, and usually after the column, you'll have a detector, for example, a UV detector, before the sample flows into the X-ray beam, and you collect SAXS data. So why do we want to use SEC-SAXS? Well, you've heard from Richard and also Mikhail about several difficulties with SAXS. Like, for example, you have to have this good buffer match. You're very sensitive to aggregation. You have to have a very clean sample. So SEC-SAXS can kind of solve a lot of those problems. And actually, I would say before the SEC-SAXS technique was developed, like the vast majority of my SAXS experiments ended in failure. So this technique really made the technique more robust and useful. So, all right, so pros are exact buffer match. We can remove aggregates. We can confirm that our protein is monodispersed. So if you did a purification in the lab, you do SEC-SAXS and you get the same elution profile, it's a good sign that your sample survived the trip to the synchrotron. It can also, in some cases, separate mixtures, for example, monomer and dimer mixtures. And then the final thing is that there's other computational tools we can apply that you can't apply to just a single scattering curve. So because you have this large data set, you can apply tools to separate peaks even if they're overlapping. So I'll say more about that later. So the disadvantages, though, this usually uses more material than a standard SAXS curve, a single SAXS curve. Because the column tends to dilute the sample. So you have to load a larger amount to get the same amount of signal. The buffer condition that the sample is in is sort of fixed for the experiment because you have to equilibrate the column in your buffer first. So if you wanted to titrate a ligand or something like that, it would be very difficult. Also, the protein concentration will vary during elution from the column. I think Richard showed you an example of a structure factor, so interparticle interference. So if you have some sort of concentration-dependent protein-protein interaction, that might show up because you have this change in concentration. And then finally, you're flowing, you've got the X-ray on continuously, you're really cooking the sample, and so sometimes radiation damage can be a problem. Okay, so I'm just going to take a step back really quick and tell you about size exclusion chromatography. Most of these images I stole from the GE Healthcare Life Sciences Manual. I encourage everyone to read it. It's a good resource for understanding chromatography. So this is the basic chromatography setup that I showed before, where you have a pump, you have a sample injection loop, you have a column. And the real important thing about this is the column. So what is inside that column? If you zoom in, what it consists of is basically a tube. So you're flowing from top to bottom. Inside the tube are beads, and there's running buffer. Okay, so inside the beads, this is an example of superdex type column. Basically, you have this cross-linked agarose and dextran matrix. And the important thing about it is that there are pores, and the size of the pores is fairly well defined. So if you have a small, a protein of a certain size that can go in the pores, anything larger than the size of the pore is excluded from the pore. And the reason this can give you separation is that the objects that go in the pores can explore a much larger volume as they travel down the column. And that means that they run more slowly. And the objects that are excluded from the pores travel quickly. So in this diagram, you have orange, red, and yellow proteins. And then if you were to look at how they were running through the column, you would see that over time, they split into bands where the red thing is running faster and the yellow thing is running slower. Okay, so things that run more quickly elute first, and things that run more slowly elute last. So you have to kind of mentally convert speed to time when you're reading a chromatogram. This is what a chromatogram looks like, typically. So time is going from left to right, or sometimes it's shown as volume, elution volume. And then usually a setup will have like a UV cell to measure absorbance. Proteins and nucleic acids absorb in the UV. So, right, so on the first peak that comes through on the left here, this would be like the high molecular weight things. Peaks in the middle would be of intermediate molecular weight. Proteins of low molecular weight would elute on the right-hand side. And a column will basically partition based on how fast things run. But you can assign a number, for example, between 0 and 1, where 0 is the largest object and 1 is the smallest object that the column can separate. So this is called partition coefficient. One thing that's commonly done is you can make a calibration of your column. So you could buy, for example, a set of calibration standards like this set and run it through the column, and then assign each of these peaks a partition coefficient and make a plot. So this plot shows partition coefficient versus the logarithm of the known molecular weight. And for most of these size exclusion columns, they give you this kind of exponential relationship between separation, or partition coefficient, and molecular weight. So this gives you a sort of rough calibration of the column. And so something to say about this is that you can use such calibrated columns to measure molecular weight, but it's typically not very accurate. And that's because this sort of assumes that the protein is globular. And actually, if you're running, for example, nucleic acids, the molecular weight you get from its partition coefficient of nucleic acid could be off by as much as a factor of 4. So it's really not a good way to measure molecular weight. Thankfully, there are other ways to measure molecular weight, and SAXS is one of them. Okay, so you may have used SEC to do purification in the lab. It's very common as a final step in purification. When you do that, typically you're using like a prep grade column, which is massive. That's not what we use for SEC-SAXS. We use analytical scale columns. Those have a pretty small volume. It's usually between 2.4 and 24 milliliters. And the reason we do that is because we want to minimize the amount of dilution of the sample. So SAXS requires fairly high concentration of protein. So we use a smaller column. So that minimizes sample, also minimizes the time it takes to do the experiment. We're usually using these either 150 to 300 millimeter long columns. The 150s will run faster, the 300s give you better separation. And then depending on the beamline, the facility, there are sort of two different brands of column that I've seen. One are these sort of polymer and sugar matrix columns made by GE, so SuperDex or Superose. The others that I've seen are Shodex columns, which are SiO2 beads. These Shodex have better resolution, but they don't tolerate pH less than 7.5. So there's trade-offs there. The SuperDex are much more sort of have much better chemical compatibility. Okay, and then the other important choice is exactly what kind of media you have. Essentially, what are the pore sizes in your bead? And different columns are rated for different molecular weight ranges. So you want to choose an appropriate column for the sample you're looking at. I think people typically start with like a SuperDex 200. This is a pretty standard molecular weight range, but then you might need to go up or down depending on what your protein size is or what the contaminants look like. Okay. So here's, I just want to give a more concrete view of what the experimental setup looks like. So this example is from CHESS, although it's fairly old. So the CHESS setup doesn't look quite like this anymore. But basically you have a commercial fast performance liquid chromatography machine like this ACTA, where all the pumps and valves and columns are integrated into one box. And then you have the SAXS beamline. Here's what it looks like. Whoops. Don't know why that happened. We'll try it again. Here is SAXS beamline, and then all you do is connect these two together with a piece of tube. So that's literally what you do. Just flow right from the column into the X-ray beam. And usually there will be some kind of in-vacuum capillary flow cell that's used. So this is a picture of one we've used at CHESS in the past. And then the procedure for data collection is also pretty simple. You just frame the camera continuously, the SAXS detector continuously while you're eluting, and you collect these three-dimensional data sets that I showed before. So our intensity versus time versus Q. And then the next step is to analyze that data. So I'll show you some basic analysis, and I'll attempt to do this with a live demo as well. So first thing, okay, so this is an example data set that I collected now in 2015, a long time ago, but I just did something really stupid, which is I took BSA powder and I dissolved it in a random buffer and injected it and sort of wondered what I would see. So this is what the UV trace looks like from that BSA powder. And you see kind of a main peak, but you also see a shoulder on the left-hand side. So there's the main peak. It's basically the BSA monomer. The shoulder is some kind of dimer, trimer mixture. Okay, and you can also say, okay, what do the scattering curves look like? You have mostly this background scattering, which is from the buffer, but then you also see some scattering from the elution. Okay, so now what people typically do is they make something called the Sax Chromatogram. This is just a plot of the mean SAXS intensity versus time or versus elution volume. Okay, and you can see this looks a lot like the UV trace I showed you before, but there's some features that are slightly different. So one of them is you see this negative peak here that I've labeled buffer mismatch. And this is because the buffer that the sample was in when I injected it was different from the running buffer. It had a different amount of salt. And so you can see this as well in the conductivity trace. So the FPLC we were using had a conductivity sensor, and there's a blip in the conductivity. It's telling you there's a buffer mismatch. So the reason I point this out is because we always tell people to use the buffer from the beginning of the elution for your buffer subtraction, not at the end, because this buffer mismatch can be an issue. So this region, this buffer from the sample is equilibrating with the running buffer as it elutes, as it goes through the column, and so this is the proper buffer to use for background subtraction. Okay, so you can do background subtraction, and then do Guigné analysis. This is done automatically. So basically, you're fitting that Guigné region for every single curve in elution, and you can make a plot of RG versus time, and also the forward scattering I of 0 versus time. Yeah, and what you see is, sort of this hypothesis is basically right, that there's something large, that's coming off before the main peak. And, RG is fairly constant in this region, so we can just average these scattering curves. Okay, so one reason that the Sax chromatogram is kind of the first step in data analysis is you can often catch issues. One issue that occurs sometimes is radiation damage, and in particular, there's this issue of capillary fouling, where essentially the X-rays will cause protein to stick to the sample cell, and it will sort of build up over time and cause a background to change. This is like the worst example of capillary fouling I've ever seen, but it illustrates the point. So here's, if you look at on the left-hand plot, if you look at the, I think it's this blue, yes, the dark blue is the radius of gyration. So it goes up in the main peak and it goes down, but it never comes down to 0. And then similarly, this this light blue, oh, sorry, I got it backwards. Light blue is radius of gyration, dark blue is the forward scattering. Yeah, so basically they never come down. That's just because you've got all this gunk building up on the capillary, and that gunk scatters like crazy. But in this particular case, they found that all they needed to do was switch the buffer from heaps to phosphate, and this radiation damage issue went away. So maybe the lesson from this is phosphate is a bad buffer to use, although I've heard people say that phosphate is fine. But I guess the point is that you want to be able to hopefully diagnose your problems and respond to them to get the best possible SAXS data. Okay, so I'm going to attempt a demo here, and we'll see how this goes, because I have to switch windows. Okay, so you should see my raw analysis window now. So for this demo, I'm just going to load up some SEC-SAXS data from xylanase, which is just kind of a standard protein. And the reason I'm showing this data is because it's not perfect. So when you look, it's got kind of one main peak here. This is the Sax chromatogram. What's being plotted is the total integrated intensity. So it's got a main peak, but there's this sloping background. And this sort of thing can happen. Sometimes there's drift in the normalization of the SAXS instrument. Sometimes the column hasn't been equilibrated quite well enough, and so the buffer component is changing slightly over time. But this sort of minor drift is sort of correctable, and I'll show you how that's done in raw. Unfortunately, when I open this pop-up window, I'm going to have to share the pop-up window. So let me stop sharing, open the pop-up window, and we'll share again. Okay, so this is just what's called the liquid chromatography analysis tab in raw. So I said the first thing you want to do is select a buffer region. So we're going to select a buffer region here. In raw, you can just pick it. So I'm selecting from before the peak, as I've said. It's kind of the proper region to choose. So we set buffer. It complains. You can ignore that. Okay. All right. So that, it's subtracted now. So total intensity is sort of 0 here, but we still have this baseline drift problem. So raw actually has the ability to correct for that. I'm going to use a linear correction because you can see it's quite linear. And I'm just choosing some regions here to average. Probably want to average more than that. Let's see. And this will be too much. Oops. Okay, so we're going to set baseline. Great. So yeah, raw was more or less able to correct for that baseline drift, which is nice. So now, if we want, we can select a region of this curve that we're interested in and plot the scattering. So I'll just select sort of a region here. Let's see, pick. So I'm just going to average over the main peak. And maybe I'll do another one where I look at this gunk on the left. Let's see. So this is probably some aggregate. And then maybe we'll look at something on the right as well. All right, so now I'm sending them back to the main interface. And not sure why it's so slow. Yeah, okay. Now I need to share my main interface again. Okay, so this, oh, well, it only gave me one, unfortunately. I'm not sure why. Yeah, okay, well, we only got one. But in any case, you can see that we've got, on a log-log plot, we have a nice scattering profile, and we could do further analysis on this if we wanted to.
Just a comment, if I may.
Mm-hmm. Sure.
But for the SIBYLS users, most likely also with this other beamlines, we doing this processing for you, so you're obtaining besides the whole frames and also the HDF5 files, what can be directly plot in raw. We also give them the final merge data. So, but it's good to check our works in case you want to do that this way.
Yeah, I think that's I think SIBYLS is in some ways unique that they do a lot of processing for you. I guess BioCat I've heard as well is doing a lot of that. If you come collect data at CHESS, you have to do it yourself.
Yeah, we're not pipelined yet. But as soon as Jesse has worked out the we'll adopt his technique, I think, and do more of that.
Yeah, I mean, I think when the experiment goes well and the data are very clean, this analysis is fairly straightforward. But I'm going to show you lots of examples that are really messy, so you'll get a feel for when you need to kind of dig in and do things yourself. Okay, so that's I will share my slides again.
Not to give the impression, I'm sorry to barge in, Steve.
That's fine, yeah.
That not to give the impression that SAXS goes wrong a lot of the time.
It goes wrong.
Just to let you know what it does.
Yeah, a lot less often than it used to.
I think we lost Steve.
Oh, did he? He just dropped out.
I think he clicked on something what he's supposed to not click because there were so many clicks in
I'll be back in a
between.
minute.
Because now he was probably trying to talk about more complicated. He's back.
Sorry about that.
No.
No, my Zoom got overwhelmed. Okay. Let me try sharing this again. All right, great. So I've shown you basic analysis. I'm going to show you now some more advanced techniques. So these are actually sort of general techniques you can use for SAXS that are based on the idea that SAXS is telling you about mixtures of things in solution. And so to kind of illustrate this, I'm using this fish analogy. So, when you have mixtures of things, basically the scattering you observe is a combination of scattering from all these different objects, and you want to use techniques to try and unmix the data and learn about the scattering of the individual components. So the theory of this is basically that if you have a very dilute solution, here dilute means that the fish are not interacting with each other. So you can kind of approximate the mixture as just independent yellow fish plus independent green fish. So if that's the case, then the scattering profiles also add linearly. So it's just the scattering of the mixture is the scattering of the yellow parts plus the scattering of the green ones. So there's no term that's telling you about interactions between the two. So if that's the case, you can use methods from linear algebra to deconvolve these mixtures. And so the techniques I'll tell you about are just based on linear algebra methods. First method is singular value decomposition. This is applied in SAXS field all the time. I think the first example was 1996 by Chen and Hodgson and Sebdoniak. And the basic idea is you put all of your data into a single matrix A, where the SAXS profiles are like columns in this matrix. And you perform the singular value decomposition, which is automatic. And it decomposes that matrix into the product of three other matrices, U, S, and V. The matrix U has columns that are kind of like scattering profiles. Matrix V has columns that are kind of like elution profiles, if we're talking about SEC-SAXS. And then this matrix S is diagonal, and along the diagonal are the so-called singular values. These are just positive numbers, and they're usually ordered in order of largest to smallest. And they tell you basically how important each of these component vectors are to in reproducing the entire data set. So in this particular case, there are three very large singular values and then a whole bunch of small ones. And this is telling you that there are sort of three linearly independent components in your data set, and then the rest of this is just needed to represent the noise in the data. So this is useful information. So we have a SAXS profile. Maybe there's only a single elution peak, but from the SVD, we notice that there's actually three components in there, and so we have to use some more advanced analysis. We can't just average this peak. So I'm going to show you hopefully a demo. Let's see if I can get this one going. Share. All right, so this particular data set is one I collected a long time ago. It's actually the one that kind of motivated us to use SVD for this purpose. Okay, so here's the data set. It's from an enzyme called phenylalanine hydroxylase, which is a tetramer. And it has some interesting allosteric transitions we were trying to look at. And this SEC turned out to be really important for getting the single tetramer scattering, because it was always kind of interconverting with some larger oligomers. And I'm going to go ahead and launch the SVD window, which means I'll have to change my screen sharing again. Okay, so here's SVD window. So what RAW has done is it's applied SVD to that entire data set. And on the bottom right panel, we're plotting the singular values. Oh, sorry, no, the top right panel is the singular values. You can see there are 1, 2, 3 sort of largish singular values and then a whole bunch of small ones. So there are 3 components needed here. So one thing we can do in RAW is we can choose which frames we're using to calculate the SVD. And you can see on the right-hand side of our elution peak is a little bump. That's another buffer mismatch peak. So we can just remove that. So we're going to use frames up to say 310. So we remove that bump. Now, if you look at the singular values, we only have 2. So this is good. So one of those components was just that buffer mismatch peak, and we've removed it. So now the question is, okay, so we've got one main elution peak, but there's 2 singular values. So it's actually kind of 2 overlapping peaks. So we can kind of like test this idea by selecting different regions. So now if I select, for example, only the right-hand side of the peak, so everything after index of 200, now we only have 1 singular value. Does that make sense? So what I'm showing here is that basically on the right-hand side of this peak, this is all basically a pure single scattering species can be represented by one scattering profile as just being rescaled. But if I include the whole peak, there are two singular values. That means this is really two overlapping peaks. Okay, so this is kind of how SVD can be used. I mean, we could say, okay, we're just going to take the right-hand side of that peak and average it. But we can also use more advanced analysis methods. So I'll tell you about what those are. I need to start this again. Okay. Right. So to kind of motivate that, I'm going to show you a more complicated data set. I made this data just for the purposes of testing algorithms. So I just mixed a whole bunch of chromatography standards and ran them all on SEC-SAXS. So we got lots of peaks to work with. Now, what happens if we do SVD in real time as things are eluting from the columns? That's what I'm showing here. So on the left, we're watching Sax profiles come in, and on the right, we're plotting singular values, sort of in real time. And what you'll notice is that every time a new peak comes through, there's a new singular value pops up above the noise. So there's 1, there's 2, there's 3. There's another 4, 5, 6. Okay, so this should give you the idea that we can use this kind of singular value decomposition as a function of time to tell us about when new species are eluting. Oh no. I'll try that again.
You can share the whole screen.
You know, I can. The problem is my monitor is huge, so everything
Okay.
would get shrunk really small. Okay. So this is the basic idea behind a technique called evolving factor analysis. It's basically that movie I just showed you, but put into a single graph. So we're plotting the singular values as a function of time or as a function of frame number. And as a graph, you see these little blips. Basically, every time a singular value pops up, you can draw a vertical line, and we call these inflection points. So the inflection points occur whenever a new component elutes from the column. You can also do the same thing as if you had done the experiment backwards in time. And what that tells you are when components leave the scattering volume. And what's cool is now we can match up forward and reverse, these forward and reverse plots, and just match up when where the inflection points meet. And because we're using SEC, size exclusion, we can use a first in, first out principle. So whatever comes in first must leave first. And then based on that, we can basically assign different peak ranges. And once we have those peak ranges assigned, you can actually just do an optimization. We call this rotating the basis. Basically, you're doing a least squares minimization of each of these components outside of its peak region, and forming new linear combinations of these basis vectors that SVD gave you to get basis vectors that are more or less physical. And so this is cool. So what we've done is we've taken our data set, which had a whole bunch of peaks, some of which were overlapping, and we've automatically separated them out into individual components. I'll show you now a demo of this using some real data. It's again that same phenylalanine hydroxylase data set I just showed. Before I share my screen, I'm going to launch the evolving factor analysis window. Okay. There we go. So what you're seeing now is the SVD. So SVD is kind of the first step because you want to choose the number of singular values to look at. So we chose 3. Now we're doing calculating the forward and reverse evolving factors. This takes a little bit of time, but there they are. So on the top plot are the forward factors, on the bottom plot are the reverse ones. And raw has kind of made an attempt to choose those inflection points automatically. You can see it's done a pretty good job, but we can actually go in and adjust them a little bit. So I'm moving that blue one a little bit to the left, and then maybe this other blue one, we want to move it to the right. Okay, when we're pretty happy with this, we can just click next, and it'll do the basis rotation. So that's what it's done here. So on the top, these are the scattering profiles it's pulled out for each component. And then on the bottom right is showing the concentrations for each of those components. There's different options. For example, you can force concentrations to be positive. I usually just turn this off and let them bump up and down around 0, because sometimes it's good if you've assigned these ranges incorrectly. Sometimes you'll see negative concentrations. But this looks like a pretty good solution. One thing we also do is we look at the chi-squared. This is the chi-squared of the residual. And if we'd chosen a peak range badly, for example, if I make this orange one start a bit too late, what you'll see is there's a big jump in the chi-squared here. It's telling you that your assumptions here are not allowing the algorithm to fit the data. So it's really important to choose these. It's really important to choose these peak windows well. And yeah, the chi-squared can kind of be a guide, so kind of adjust a little more and see if you can minimize it better. Okay, so that's basically how EFA works. And when we're done, we can go ahead and save these profiles to the save those profiles for further analysis. So actually, this technique was absolutely essential for us to isolate that scattering from the tetramer. Although in retrospect, we could have just used a better column, I think. Okay, so let's go back, dive back in. Let's see, I think I was supposed to end around 6:15, is that right? I guess we got a little bit of a late start, but I don't want to go over if there's a time crunch here.
You definitely have, like, Craig is okay with that, at least.
Yeah. I mean, yeah, 6:15 your time was the official
But
question.
definitely have.
I can cut my talk a little short.
Okay, yeah. So I think what I'll do is I'll show these advanced applications really quick, and then I'm actually going to maybe go through a little bit of the Regals method, because I think it's kind of it will be interesting to this audience, and then show a quick demo of Regals for one anion exchange data set, just to make it more concrete. Okay, so let me share. Oh boy, I got the whole thing. Okay. All right, there we go. So what is SEC-SAXS good for? One area where it's particularly useful is studying membrane proteins. And the reason is that often when you are preparing membrane proteins, you're using a detergent to solubilize them. And this can cause a lot of problems for conventional SAXS. In particular, you're operating close to the critical micelle concentration, you can have free micelles forming spontaneously, and that's contributing background. So this particular study, they looked at aquaporin that was stabilized using DDM micelles, and SEC-SAXS was used to kind of equilibrate the detergent concentration and to separate the protein in micelle from the free micelle, and also to produce an accurate buffer match for subtraction. So this would have been impossible to do using a conventional data analysis or data sample preparation. Getting that buffer match would have been really difficult. Okay, another thing people are doing is coming up with clever ways to get to reduce radiation damage, and this is this CoFlow setup has been implemented at several beamlines. It was first described by Nigel Kirby at Australian Synchrotron And the idea is you basically don't ever let the protein touch the walls of the capillary. So I said that this capillary fouling issue is because proteins will stick to the walls. And so when you envelop the protein in this sheath of buffer, it's never able to do that. And they're able to show some really good performance on this system. They can crank the flux way up and get very good signal to noise. So you'll probably see these CoFlow setups being adopted more widely, particularly as synchrotrons are upgrading and getting higher flux.
Steve, just a short comment also, if you don't mind. Also, we have experience when we running a couple of protein standards prior to samples, the protein standards producing the clunch on the capillary. It's definitely increased a little bit the background, but for the samples are less or more, we don't see so much the capillary falling afterwards because the first layer kind of protecting the capillary from additional aggregations on the samples on the capillary.
Yeah, that's interesting. I know people.
I know there are beamline scientists around. I'm supposed to not say it loud and clear, but it seems it works, especially running BSA for each users.
Yeah, that's interesting.
And the BSA is a high concentrations, and so they really producing this protective film on the surface of capillary.
Yeah, that's an interesting observation. I know that people who do single molecule fret will often use like BSA to passivate their cover slips. I think it's similar, must be similar mechanism. It's like highly charged. Cool. Okay, so the other thing a lot of beamlines are doing now is SEC-SAXS also with inline multi-angle light scattering. So multi-angle light scattering can give you a much more accurate molecular weight estimate than SAXS does. So it's very complementary. This picture I've shown is from BioCap, but many beamlines have the same instrument. It's made by Wyatt. And this is an example of how that looks like. So basically you get a very accurate readout of molecular weight across your elution peak. And so you can be very confident whether that's a single species or there's some kind of exchanging oligomers, for example, that would give you a changing molecular weight. And this is, very powerful technique. There are some more requirements for sample preparation and choosing column media and things to be compatible with this MALS setup. The other thing people are doing is using other kinds of chromatography, so not just SEC. Our group, the Ando group, has played around with anion exchange coupled SAXS. This was an example where we're looking at ribonucleotide reductase from B. subtilis, and our collaborators had done this AEX experiment in the lab and shown that there were two sort of peaks that they observed, which they labeled holo and apo. It had to do with whether nucleotides were bound. And they wanted to know structurally sort of what these were. So we just did the same experiment, but coupled to a SAXS beamline. And the challenge for this data is that because you're eluting with a salt gradient, so going from 100 to 500 millimolar NaCl, your background is changing continuously during the experiment. And so it becomes very hard to do background subtraction using any kind of conventional method. So we actually developed a new data processing method called Regals that I'll show you in a minute to do that background subtraction. But this is an example where the data are shown in this plot here. The holo curve is plotted twice with two different models for what that structure could be. These are two different structures that were derived from crystallography, a canonical dimer and a non-canonical dimer. And in this case, the SAXS strongly suggested we had this non-canonical thing, which actually we were able to confirm later using cryo-EM. So this was a pretty cool application of SAXS. Okay, so I'm just going to briefly summarize what I've told you so far before we move on to Regals. So SEC is really great. It can separate molecules based on size, and it removes aggregates, separates oligomers, provides a good buffer match, and it just increases, I think, your overall confidence in your SAXS data, which is important. And it typically requires extra sample and extra time, so you want to plan for that. And then you also need to take care to choose, your proper column, set your experimental variables well, assess your data quality, hopefully in real time, so you can make changes if something goes wrong. And then once you have the data, you can now apply these powerful deconvolution methods that I've showed you, like SVD and EFA. And there's, I think, a lot of people working on new methods. So it's kind of an exciting thing, and you want to pay attention to what the latest technical developments are. Okay, so I just want to tell you a little bit about Regals, because it's very similar to EFA in some ways, but it's sort of more powerful and more general. So we are motivated to deal with this because EFA can struggle sometimes when you have highly overlapping peaks. This is an example that I've just simulated some sort of difficult data where we have tetramer, dimer, monomer, which are just spheres, and the peaks are heavily overlapped. So we get like a single peak coming out of our SEC-SAXS, but actually it's 3 peaks overlapping. We run evolving factor analysis. It finds the peaks okay, but then when we do the basis rotation, it's not able to recover the monomer component correctly. So you see this yellow bump. It has like bizarre looking intensity, and it's because it's like a mixture of some monomer and also some negative dimer or something like that. Okay, and the other thing we were worried about, of course, is this anion exchange chromatography. We couldn't use EFA in this case either, because of that sloping background. So that causes a change in the number of singular values that's not really related to when proteins are coming in and out. And again, if you do evolving factor analysis, you get some peaks, but it doesn't work when you try to do the rotation. Okay, so so how do we solve this problem? We developed this method called Regals, and the basic idea is we're going to take basis vectors. We're going to try and add some extra information when we do the rotation. So we're going to design these basis vectors as parametric curves that kind of express what we mean by physically meaningful solutions, meaning we want to have concentration peaks that are sort of smooth and that are confined to a certain range. We also can impose restraints on the SAXS curves. We can say the SAXS curves ought to be smooth, or the SAXS curves ought to have a P of R function with a certain maximum dimension. So for example, if we knew that something had a D max of 100, we could say, okay, this component has to be like, have that restraint. And then the user basically can mix and match these different restraints to describe their experiment, the prior information they have. And then we just refine the whole thing globally. Let's go kind of quickly through this since I don't want to use too much time. But basically, yeah, there's different kinds of basis vectors we can choose. We can have simple or smooth. Simple just means no restraints. And then in real space, you can also have these P of R functions, which is kind of a unique feature. And it's a difficult nonlinear optimization problem to solve. And the method we use is called alternating least squares. So regularization plus alternating least squares is where the Regals name comes from. And it's iterative. So you start it off on with some initial conditions, and you let it evolve until it finds a good solution. So on that example I showed before, on the left here is the poorly resolved peak from EFA. If we now say that, okay, the concentration peaks have to be smooth and confined to certain regions, we're now able to recover those components much better. And on the right here, I've shown when we use the P of R information as well, that does an even better job. So removes more ambiguity. I'm going to actually show you the application of Regals to AEX-SAXS. We'll do this as a live demo rather than slides, if we can. So before I share my screen, I'm just going to load up that data set. Okay.
In between, I'm really glad, Steve, you really explained it better than everybody can do in our group. So highly appreciate this slides, especially we heavy using SEC-SAXS for the users. They trying to do structural solution determination.
Great, yeah. So this is that data set, the SEC-SAXS data set that I showed. This is in raw. This is the first step. It does basically an SVD of your data, and on the left you can see this is the integrate total intensity. This is crazy sloping background, which is coming from that salt gradient, and you can see there's four large singular values. It turns out that I'm not showing this right now, but it turns out that two of those singular values come from the buffer component. So there's actually kind of a nonlinear scattering that happens due to the salt. It's not just a simple linear background. And that requires two singular values to describe. So it's actually a pretty tricky data set. So raw actually has some preset methods, sort of defaults for regals, which is nice. So we can choose the anion exchange SEC-SAXS preset. Go to the next page. So here's where we define basically our model, what we're going to use to describe these peaks. And there are a lot of different options, and here I'm not going to explore. Basically, the process of fitting a Regals model involves a little bit of fiddling around with these parameters. But since I've already done this before and I know more or less what works, I'm going to show you that rather than the fiddling around. So we're going to leave the SAXS profiles alone. We're not going to mess with those at all. We're only going to impose restraints on the concentration peaks. So we're going to make them smooth. So there are 4 components, make them all smooth. And then, well, it's important to set this range here. So this peak 0, I'm going to say is a background component. So we'll let that be the full range. And similarly, this one I'm going to have as a background component as well. Component 2 will be the first peak, first protein peak. And I'm looking at my notes here to see the range, 730 to 1270. Okay, and then the second peak, similarly, you just set the range. So we're going to go 1150 to 1600. And tell it to be 0 at the endpoints. Okay, so now all we have to do is hit run. So we run Regals, and it spits out an answer. So you can see it's minimized the chi-squareds really well. In the lower right are what those concentration profiles look like. So blue and orange are the ones describing the buffer. Green and red are describing the protein. And it's interesting, you can see that the actual protein elution peaks are very bizarre looking. And that's, it's partly because this anion exchange chromatography is not quite what we're used to looking at with SEC. So it's separating based on charge, and this particular column can separate, it's sensitive to a single charge on the protein. So a difference of 1 electron. So anyway, it produces weird looking peaks sometimes, but the cool thing about this is we're able to get, well background subtracted SAXS profiles. Okay, there's other things you can mess with. For example, this lambda parameter controls how smooth the peaks are. And the defaults are usually pretty good in raw, but since we want the backgrounds to be really ultra smooth, you can set this to be a large number, say 1 times 10 to the 8th, and rerun Regals. Now you should see these background components are much smoother. So sometimes that's important to do. Okay, that's it for that demo. I'll go back to my slides and just show you really quick some of the other things Regals can do, and then we'll be done. Okay, so I showed you this SEC-SAXS example, or AEX-SAXS. I was also going to show you, oh, there's the demo. Okay, so the other thing we can do is analyze ligand titrations. This is an experiment we do a lot in the Ando lab, where we have an enzyme that has some allosteric conformational change. And so you can titrate in the allosteric effector and see the change in the scattering profile, which is really cool. And we try and understand that. But this particular titration had some issues. The change we were expecting was at mid Q here, but we also saw a change at low Q, and that low Q change was related to an increase in the average molecular weight. So there was some kind of oligomerization happening that was unrelated to the allosteric mechanism. It happened outside the physiological range of the ligand. So there was this sort of ugly mer forming, and we had to figure out how to separate those two effects in the data. And so it turned out Regals was able to do that. And all that we did is we said, okay, this molecular weight change must involve a P of R with a very large Dmax. So that's what's the orange thing is showing. And then the two tetramer species, the inactive and active species, those were given the same Dmax of 130, and then Regals was able to pick that out. Okay, and it also works on time-resolved data. So we've analyzed some publicly available time-resolved data sets. So this top one is a rapid mixing, stop flow mixing experiment that was done on a ATP binding protein that undergoes monomer dimer transition. And also we've looked at some laser T-jump experiments that were done by Michael Thompson at BioCat. And this is sort of like sort of state-of-the-art crazy SAXS experiments people are doing. So I showed you the Regals implementation in raw. Actually, that was Jesse's doing. So our lab's Regals code is available on GitHub, and it's actually, if you want to use some more powerful features in Regals that aren't available in raw, this is the place to do it. There's two versions. There's a MATLAB version and a Python version, and the code is run in notebooks, so Jupyter notebooks in Python and live notebooks in MATLAB. So you sort of have inline code and documentation and output. And there's a bunch of example data sets there too you can play with. So check that GitHub out if you're interested in learning more. And at the very end of my slides, I just have a bunch of information tables. I think you'll have access to my slides, or I'll give you guys all a copy so you can see these. But basically, there's some troubleshooting and references and links to more information. All right, so that's it for my talk. I'm happy to take questions if there's time.
So I had a question about the deconvolution with SEC-SAXS data.
Mm-hmm.
So if you have overlapping peaks, is the result coming out of the deconvolution going to be good compared to like if you optimize the chromatography protocols to separate them more?
Yeah, that's a great question. I'd say it's always better to have good chromatography data. It doesn't always work, and you know, part of the problem with SAXS beam time is, you know, you'll do your experiment and maybe you can't come back and do it for a long time. So you have to just make the best of the data you get. And so that's kind of what motivated it. Sometimes we've actually found cases where we really can't separate things, that they're interchanging on the time scales of the separation, or they're just too close in molecular weight. So in that case, you sort of have to rely on the evolving factor analysis or the SEC-SAXS. So as long as they're not too overlapping, the results are usually unambiguous. They can become ambiguous when the peaks overlap too much. So in those cases, that's when we sort of rely more on Regals, for example, to bring in extra information that's coming from outside. That's not, you know, we're not relying only on the scattering data to tell us what's happening.
Just a comment, Steve, like before another questions. Also, you probably may check the multi-angle light switch ring if you collect that to see if the overlapping peaks or completely broad peak have indication of different molecular weights across the peak. If not, you really have a hard time to using any Regal or EFA if the molecular weight seems to be something in between monomer dimer example.
Yeah, that's a good point. I've actually never used multi-angle light scattering. So, but if you have access to it, it's great. And I think, you know, we're interested in adding multi-angle light scattering data to Regals, for example. I mean, you could think of ways of adding it if they're synchronized exactly in time, but we haven't done that yet.
Steve, what's the minimum number of curves you would think Regals would want to deal with?
Oh, that's a good question. I mean,
We do
I've
iteration
never really,
series, right?
yeah, I've
So-
never pushed it. I mean, I think whenever we do a titration, the grad students really suffer because the sort of in the lab we always try and really oversample that titration just to get a lot of data points. So it's usually like 10 to 20 scattering curves.
But it will work on 20 curves.
Yeah,
Rough.
20 curves is fine.